Greetings students, welcome to one of the final topics of this course on algorithmic thinking in bioinformatics. Today we are going to look at how genomes evolve via large scale evolutionary events such as large scale segmental rearrangements and shifts of segments of chromosomes or genomes. Okay. And these large scale mutations can give you a very different view than what we have seen in the course so far on gene level mutations, where you look at single genes or single proteins and then understand what are the mutations that transform the gene of one species to the same gene in another species, right. And because we are looking at these large scale mutations, not point mutations or insertions or deletions within a single gene or within a single protein, the algorithms we are going to use would also change. And so there also we need to use a different style of thinking. Instead of using dynamic programming, for instance, to align two sequences to understand what are the minimum possible mutations that will transform one sequence to another sequence, we are going to use combinatorial algorithms and some form of graph based algorithms to understand how to transform one genome to another genome. Okay. So, the problem is new, the algorithms are new, and let us go ahead and see what it is. Okay. The style is going to be the same, though. We are going to start with the biological question as in the other topics in this course, and in this particular case the question is how do chromosomes or genomes evolve via these large scale rearrangements, and then how do we transform a mouse into a human genome. So, that is just an example application of this question. So, once you understand how chromosomes or genomes evolve via these rearrangements, then we can use that to figure out the minimum possible rearrangements that you need to do to transform one species genome to another species genome. So, in this example we have taken a mouse genome and a human genome. Okay. We will take that as a running example in this session. And then if you look at the associated computational problem, we can map this rearrangement problem to a certain kind of a puzzle. We have a permutation of or ordering of certain genes in one species, and then you want, and the same genes are in a different order in another species. So, you are sorting one permutation which is an ordering of genes, and to get it into another permutation which is another ordering of genes in another species. Okay. So, the computational problem is sorting a permutation of gene orders, right, between two species genomes. So, sometimes this ordering of genes I also call it as synteny blocks. We will get into the details a bit later. Okay. So, for now you can just assume that synteny blocks are just the ordering of genes. Okay. So, there are different gene orders in different species, and how do you get from one ordering to another ordering, and that is the computational problem of sorting one permutation to get another permutation of the same set of objects. And the algorithmic solution, as I said before, is going to be somewhat different also from what we have seen earlier in the course. It is going to be some combinatorial algorithms that are based on a data structure called breakpoint graphs that captures the structure of a permutation, how close a permutation is to the identity permutation. Okay. And again the details may look a bit mysterious now, but we will clarify it as we go along. Before we get into the problem, let us also just remind ourselves why a genome level study is important and complementary compared to single gene level studies. Okay. So, recall that this genome is the total DNA content of the cell, right. It contains both the nuclear DNA as well as the mitochondrial DNA. We will focus mostly on the nuclear DNA for today's session. And then in humans, for instance, the nuclear DNA is organized into these 23 pairs of chromosomes, 22 pairs of autosomes, and then you have this gender chromosomes X and Y, right. And then one of the chromosome is blown up here, right. And then you know that there are these genes which are in different substrings of these chromosomes, right. Here there could be one gene, here there could be another gene, here there could be a third gene. So, gene 1, gene 2, gene 3 and so on, right. So, within these chromosomes there are different genes located, and then we wanted to study the ordering of these genes, the ordering of blocks of genes together, rather than looking at single gene, how it evolves between different species. Okay. And the idea is that this will give you an overall picture of the genome, overall organization of the genome, the architecture of the genome in different species, and how the overall architecture or the organization of the genome has transformed or evolved from one species to another. Okay. again to drive home this point about single genes versus the whole genome. So, in single gene mutations you could have point mutations like substitutions, here T has become a C, or you could have insertions of deletions of single character or some groups of characters, we have called it as gaps, right. So, in sequence alignment you have these two sequences and you are trying to align it using some form of a dynamic programming algorithm. We called it as a Manhattan problem, right. We framed it as a Manhattan problem in an earlier topic. So, we had this alignment graph and we are looking for a longest path in this alignment graph, which was a directed acyclic graph DAG, right. And then that was sufficient, that algorithm was sufficient to transform one sequence, which is the top sequence, using a minimum number of mutations, insertions or deletions into the second sequence, okay, where these are the sequences of the same gene, a single gene across two different species. Now, we have in this today's topic, we have genome level mutations. We have whole segments or blocks of chromosomes, okay, a single genes next to each other like blocks of genes. These blocks or segments of genes are rearranged in all possible ways, right. So, you can look at a particular chromosome and a chunk of the chromosome gets reversed, right, or you have two chromosomes and then from the ends of those two chromosomes may get translocated and then their arms may get swapped, right. So, you have these large scale mutations that are happening in the genome, and how do we understand what are the sequence of large scale rearrangements that has happened between two different species, okay. And in that sense, what can we learn new about the evolution of these two species, okay. And why do we think that we can learn something new, right? Compared to gene level mutations, right. So, because these are very different form of mutation, these are not very small mutation, these are large segments that are moving around or getting flipped, getting reversed and so on, right. They can offer new evolutionary insights and also new insights into disease, okay. So, what are the new evolutionary insights you can obtain from understanding genome rearrangements? The idea is that they often occur at a slower rate of evolution than point mutations, right. Point mutations can occur a bit more faster in evolutionary time scales, but then these genome rearrangements, because they are large scale rearrangements, it will be deleterious if it just happens very often, right. So, these happen rarely, which means that you can understand species which have diverged very long time ago, right, because you can still understand about those past evolutionary events. If something is changing very rapidly, then if two different species have diverged a very long time ago, like 75 million years ago or more than that, like 150 million years ago and so on, right. So much evolutionary time has passed that the current day sequences would be almost unrelated to each other, right, in terms of point mutations or single gene analysis. But in terms of genome rearrangements, because that happens at a much slower rate, we can still learn about the evolutionary transformations of two species that are whose common ancestor is way down in the past, okay. On the other hand, right, in the opposite end, there are some cases, example in plant mitochondrial DNA or mammalian X chromosomes, right. So, gene reorderings can happen, right. The order of genes along a genome, maybe in one genome there are 3 genes, let us say gene 1, gene 2, gene 3. In another genome it may be not 1, 2, 3, but it may that the second gene is first. So, it may be gene 2, gene 1 and gene 3, okay. There may be different orderings of the same genes in two different species, and that can happen even if the point mutations are happening rarely, okay. In certain cases like the plant mitochondrial DNA or mammalian X chromosomes, the evolutionary rate in terms of single point mutations within single genes are very slow, okay. They happen very rarely. In those cases, the reorderings may still happen, and then so we can understand the evolution of the X chromosomes of mammals by looking at the order of genes, how they are different between different mammals. And if two different mammalian species have similar gene orderings, then you can say they are closer in evolutionary times. If two different species have farther gene orderings, then they are farther in evolutionary time, okay. So, what these two instances tell you is that compared to gene level mutations, understanding chromosome or genome level rearrangements, those genome level mutations can give us new evolutionary insights. It does not stop there, we can also get new insights into disease, and one particular example is cancer. So, there are many cancer cell lines. If you look at their genome architecture, right, the chromosomes are all very differently organized compared to the chromosome of a normal healthy cell. So, understanding genome rearrangements that happen in cancer can tell us some new ideas into why cancer cells behave the way they do, okay. So, now equipped with these motivation, let us go ahead and delve deeper into understanding the computational problem and the algorithms for understanding the genome level rearrangements and what it tells about the architecture of the human genome. So, which regions of the human genome are fragile and can break more easily, and which ones are a bit more solid and it breaks, or the recombination happens in those particular spots less often. Okay. And so, that is an evolutionary insight that cannot be obtained by looking at single genes. Okay. And of course, it is based on the chapter on fragile regions in the human genome and genome level rearrangements in the book by Compa and Pevsner, and we thank them for sharing these slides that goes along with the book.